Uh, Thank you everyone for joining us today. It is my great pleasure to introduce Tony Montgomery, who is going to be defending his dissertation. Tony started off as a undergraduate student. He got his BA from Mānoa back in 1998 and has returned to the program. But between then, he's taken a few detours along the way. He worked as a dive master for a while. He was an aquarist at the Waikiki Aquarium. He was an aquaculturalist at Oceanic Institute. And then he ended up at Division of Aquatic Resources, which is where I first met him when he was working on black corals and alien invasive species. And he was there for about 8 years before joining the U.S. Fish and Wildlife Service in 2011, where he still works today. And in 2014, Tony asked me about grad school. And I'm pretty sure I said something to the effect of, you'd be crazy to try and do a PhD around a full-time job at Fish and Wildlife and a second full-time job as the father to two beautiful young ladies who he has two young daughters. But he took it on anyway, and despite that, he maintained straight A's through grad school. He wrote proposals that were competitive and successful in bringing in more than about $2.7 million in extramural funding. He published 6 peer-reviewed manuscripts with another that's already in review during his graduate career. His first 2 chapters of his dissertation are already published. And that third one is just waiting on this pesky defense thing to be out of the way. He's done an amazing job of pulling together a huge amount of information about the mesophotic reefs of American Samoa. And I would like to turn it over to him so that he can tell you all about that. I will ask everyone to please mute their microphones during Tony's presentation. And then at the end, we can turn it on and give him a round of applause and have some questions. After that, the public part, we'll ask the committee to stay, and we'll ask Tony some questions, and we'll ask everyone else to exit after the end. All right, so please join me in welcoming Tony. And Tony, take it away.
Thank you very much, Rob. It was a very nice introduction. I'm sharing my screen right now. So I just want to confirm, everyone can see my introduction slide to start off with?
Yes.
Okay. So let me just say thank you, Rob, and thank you to my committee for getting me to this day. It's really, truly an honor to speak to everyone today. So I'm going to try to go through my work over the last several years. I'm going to try not to rush through it too fast, but I am known to speak a little quickly sometimes. So I'm going to try to take some pauses and tell a little story about the diversity and habitat of American Samoa's mesophotic coral ecosystems and how that actually, what the kind of implications there are to the deep reef refuge hypothesis. So quick introduction of some of the things I'll be talking about today is I'm going to give you a quick introduction to the deep reef refuge hypothesis. I'm going to give you some introduction to American Samoa and a little bit about their deep reefs. I'm going to talk about the marine biodiversity of American Samoa, some of the work that I've done. And then I'm going to get into the deep reef refuge hypothesis and the data that I have and the implications of what I think the data from American Samoa shows of supporting or not supporting the deep reef refuge hypothesis. So first, if for the people on the call as not aware of the term mesophotic coral ecosystems, it's simply a fancy word for deep reefs. And they're formally defined as habitats that are characterized by the presence of light-dependent corals and associated communities. They're typically found at depths ranging from 30 to 40 meters and extending to over 150 meters in tropical and some tropical regions. So that's the fancy definition. In reality, it's not a definition that defines community assemblages or faunal breaks across depths. It's really just a practical definition, 30 to 40 meters being kind of the lower end of open circuit scuba capabilities. And 150 meters more or less is the lower limit of most light dependent corals not exactly, but close. So they certainly could range a little deeper, and the communities can vary quite a bit within that. So it's a practical definition. It's not a biological definition. And a little bit about the deep reef refuge hypothesis. So the deep reef refuge hypothesis is a bit complex, but it's actually simple in its central premise. And so essentially we have a coral reef across different depths. And so in this case, 0 to 150 meters. And so you'll sometimes hear me refer to coral reef depths. And so I'm really including mesophotic coral ecosystems, or the deep reefs, within what is also known as the shallow coral reef. So if we have a reef or a set of reefs that are across 0 to 50 meters, one of the main assumptions of this is that there's an increased amount of disturbance in shallow water compared to deep water. So as climate change may unfold over the years, and bleaching events and storm events, sedimentation events, and a long other list of shallow water impacts that we know well, one of the assumptions is those will hit the shallow reefs much harder than the deeper reefs. And so the deeper reef may provide a refuge for those species that overlap. And that leads me to the second assumption, is that there has to be species overlap between these depths. If a species is not found in deeper water, then it will not serve as a refuge, obviously. And so in this case, this little depiction I put together here, you can see the green species are the ones that are kind of in the shallow water. The blue ones are more in deeper water, and the yellow ones kind of span all depths. And so those are the ones we are interested in that may serve as a refuge. Now there's a couple others. So this was formalized back in 2010 in Bongaretz and all a paper. And so in 2019, it was some more granular detail was pulled out. And so there were things defined as what's potentially called reseeding species. So the species that actually can be a source of propagules from deep water to shallow water. And those are the ones we're the most interested in. Those are the ones that actually potentially could serve the refuge. But there's also the fact that a species is found across all depths doesn't mean it actually will provide propagules to the shallow water. So those could be more of a persistence type of a species. So they're existing, they're keeping that species from going extinct or extirpated, but they're not necessarily a recovery potential for those species. Now, obviously, beyond this gets a lot more complex, because even if a species is found across depths, there has to be reproductive ability of that species. The recruitment has to occur. There has to be resilience of those species. There has to be genetic structure across the depths. There's a time component to it. And so all of those are added complexities. I will not be talking about those today. My central focus here today will be talking about the species overlap, because without species overlap, the other aspects don't matter as much. So a little bit about American Samoa. American Samoa is a territory of the United States, about a little over halfway between Honolulu and New Zealand. It's the kind of funny looking trapezoid shape EEZ there in the South Central Pacific. It's made up of a number of different island groups. So you can see down here on the left, this is the main island of Tutuila, located here right next to independent Samoa. There's a small island group here. These are the Mānoa Islands, made up of Ofu, Olasanga, and Tau. And then there's also a small populated island up here to the north called Swains Island. And there's an uninhabited atoll down here, Rose Atoll. There are also a number of seamounts within this area. You can kind of see them here on the map. Some of these actually do come up within coral reef depths. Some of them are aphotic and deeper water. So today I'm going to be talking mostly about Tutuila, but I'm certainly happy to take some questions or provide some information on the Mānoa Islands and some of these other areas as well. But Tutuila is maybe the central focus for today's talk. So Tutuila is it's a small island for the most part. It's got a land mass of about 138 square kilometers. But the most interesting aspect about this island, it's got this very large platform that surrounds it. And so what you see here in the bathymetry is pretty much, for the most part, it's the 0 to 150 meter bathymetry around the island. So you can see it's actually quite a large platform that drops off quite quickly at around 150 meters. If we were to look at what the area that the shallow coral reefs make up of this area, you can see here in black, this is the area of the shallow coral reefs around the island of Tutuila. It's about 50 square kilometers, but mostly you'll see here, there's a little black ring all around the island, and this makes up the majority of the shallow coral reefs. You'll see a few spots here on offshore banks and patch reefs that include shallow coral reefs, and of course a big area around the offshore islet of Anu. But if we were to compare this to the depths below 30 meters, so shallow coral reefs being 0 to 30 meters, if we were to take the mesophotic coral depths and separate them up into, say, 40-meter bends, and we can call the upper the 30 to 70 meter zone, you can see here just by the black, it's a tremendous amount of morph habitat. So just based on simple two-dimensional aerial area here, you'll see that there's nearly 225 square kilometers of upper mesophotic habitat. And when I say about this, I'm not talking about the actual suitability of the habitat or the presence of any particular community. It's just simply the marine habitat across a depth range. And if we go down a little deeper to the mid-mesophotic basis 70 to 110 meters, you can see that the area drops off quite dramatically. It's mostly a smaller ring around this outer platform with some old basins within this area. So these probably were lagoons during previous glacial seawater stands. And then if we go down to the lower mesophotic, you can see there's actually very little habitat. It's down to less than 7 square kilometers. It's a very small band right along this steep slope. And if you were to do a slope analysis on this, the slopes are quite high. They're averaging about 40 degrees in slope. A couple little deeper basin areas here. But for the most part, the mesophotic coral ecosystem depths across American Samoa is mostly in the upper mesophotic depth range. If you just look at this overall, if you look at the total amount of habitat around Tutuila, 86% of the marine habitat 0 to 150 meters around Tutuila is made up of the mesophotic depths. And so this is quite dramatic when we want to think about what are our coral reefs consist of. Everyone knows the shallow coral reefs, that gets all the focus on for the management actions, the fishing activity that often goes is focused on these areas. But there's a huge amount of area out here that's virtually unexplored. If we'd expand that outside of Tutuila, the entire territory of American Samoa, it's about 79% of the area is in mesophotic depth. So this is a huge gap in our knowledge of how coral reef systems function and the implications of the communities that may exist in these areas. If we're to go a little step further than that, it's like, okay, this is the area, but what makes up of these areas? And we just do a simple hard soft bottom analysis based on existing data. We can see that more than 50% of the bottom within the upper mesophotic zone is hard bottom. And the importance about this is many times in these areas, if there's a hard structure, you're going to find coral reef communities of some sort. They might be low abundance or low densities, but there will be some coral there. There will be some type of fish there. There will be algal communities there. There will be a type of coral reef assemblage. So even if we took the 225 square kilometers and cut that in half, we're still talking about well over 2 times the amount of area of coral reef, a shallow coral reef area. American Samoa also has quite a complex and robust set of marine protected areas. And so these are the ones around the island of Tutuila. They span a lot of different areas, and they've been designed for a number of different reasons and for different purposes in mind. But there's 3 different agencies that manage these areas. So overall, there's about 56 square kilometers of marine protected areas across the island of Tutuila from 0 to 150 meters. So that's about 12.5% of the entire 0 to 150 meter depths within the territory. And so you can see the vast majority of that is the shallow coral reef with some including the mesophotic coral ecosystem depths. Some of these are actually some of these are managed by the American Samoa government, the local territorial government there. And these 4 here that have the black text boxes here show the ones managed by the American Samoa government. You can see they range quite a bit to just a little bit of mesophotic habitat of a little over 3%. up to the Whangamalu no Take MPA, which is over 75% of the bottom of that area is mesophotic depths. So this one is always an interest because of that. It was you can see here, because it extends offshore, that's why it includes a much higher percentage. One like the Pongo Pongo area here, there's not a lot of coral reef habitat within that area. It's in a fairly perturbed environment, but there are the right depths and there are reefs within these areas. There is also an area managed by the National Park Service, National Park of American Samoa. This is a Tutuila unit, and about a little over half of that is actually mesophotic depths. A lot of this area is soft bottom. There's not as much hard bottom within this one, but there is a decent amount. And then the final agency that manages the MPAs is the American National Marine Sanctuary from NOAA of American Samoa. And so you can see all four of the units they have here on the island of Tutuila have a reasonable high amount of mesophotic coral habitat, with the new island B being fairly high, again, up to 73%. It's important to note that this one was designed with mesophotic coral ecosystems in mind. And so the boundaries kind of incorporate quite a diverse set of habitats across this area. And so a little bit about the marine biodiversity of what we know so far. So there's a little over 1,000 species of fishes, and about almost a third of those have been documented within mesophotic depths. There's about 258 species of algae, and so far there's been no algae species documented below 30 meters, but there most certainly are. So this number is going to be a gross estimate of what the actual community truly is within the territory. Previous to my research here, there were 337 species of scleractinian corals listed to be present, and a little over 600 species of invertebrates. The invertebrate count is probably a very gross underestimate. So there's going to be a lot more as we focus in on invertebrate communities and document that. There's been very little on other types of corals like Anthopotherians, Gorgonians, and soft corals. There certainly have been some, but very little documentation of the species. I'm going to be getting into the coral one a little bit more later. So a number of years ago, decided to try to collect some information and data on some of these areas. And so we're able to go down and do just a small number of handful of sites, about 8 sites between 30 and 70 meters around the island of Tutuila. And so really the purpose of these surveys were to document the coral presence across sites, to try to get at how does a community compare to what we know in shallow coral reefs. And so with those surveys, I was able to find about 110 species observed, and 75 of those are able to identify down to the genus species level. And the pictures here below kind of show you some rough estimates of what those communities look like. So those communities were fairly similar as far as a habitat type, geomorphology. They're these offshore banks and little pinnacles. Sometimes they come up quite shallow into the shallow coral reef depth zone. And other times they're a little bit deeper. Some sites like this one here had actually quite high coral density and diversity. And then some other sites had a number of corals, but the densities were low. You'll also see that in some of these areas, you could tell that their coral communities were actually probably more robust than they were currently. There was a lot of observed mortality across these corals for reasons that I can't say definitive. But we could discuss that later if there's interest. Within these surveys, we were able to document that there were 3 species that are listed under the U.S. Endangered Species Act. And so those included the Cropra speciosa, Fimbrophilia paradivisa, and Pavona diffluens. I will note that Pavona diffluens there's some taxonomic issues with that. And currently National Marine Fisheries Service doesn't recognize the Pacific version of Pavona diffluens as the one listed as that was described in the Red Sea. So I included here just out of precautionary purposes, but there is some taxonomic uncertainty in this species. It could be an undescribed species. And after the surveys, I was able to determine, at least preliminarily, that there were about 6 species that were probably new records for American Samoa. So these 6 species here, they hadn't been, at least at the time that I had thought, were not documented within American Samoa. So this was kind of a good example of, within just a handful of sites, able to find potential new records. And so you might wonder, how does one verify new records? So if you go do surveys from somewhere and you come up with a list of species that were found, how do you actually say, hey, this has never been found before? Well, that's where usually you'd want to go back and you'd verify the species has been put together on a checklist. And so I decided to take on a side project that turned into a bit more than that, to develop a really robust checklist of the corals of American Samoa. There certainly has been a lot of work done on the corals there for over 100 years. And Chuck Birkeland has developed a number of lists that were quite robust. But at the end of the day, they were just lists and there were some taxonomic uncertainties. And so I wanted to kind of dive into that a little bit deeper. And so why would you really want to do a checklist? It's, taxonomy is not the most exciting thing. It's a list of names. But in reality, when you're looking particularly at communities that are not well described, when you're looking at communities that we know very little about, we need to have something to cross-reference it with. So a checklist can address biodiversity at various scales. We can look at it at the island, territorial or regional scale, or even global biogeographical scales. The names that would belong on a checklist are extremely important. The names are a biodiversity currency. So in biology, we use the Linnaean binomial taxonomic system. And so those are really important of how to track our biodiversity currencies. There's lots of different scales of biodiversity, but oftentimes we center in at the species level and try to go from there. It can provide a baseline to reference new records and the depth extensions. And so one of my goals here was that there. But depending on what information you have associated with the checklist, you can annotate it for lots of different types of peripheral and associated information. But also maybe more important than all of that is it does form a foundation for the further taxonomic, ecological, or molecular focused studies. So corals are known to be taxonomic messes. There's a lot of dispute and debate and uncertainty in what sometimes species or species complexes are. All of that matters in how to look at it ecologically. As molecular studies get underway, we're finding cryptic species and a lot of species complexes that we maybe hadn't quite realized or didn't fully understand. So having a checklist that can be cross-referenced is really the foundation for most ecological studies. And so here's a couple examples of a checklist. So the one here on the left was one of the original checklists for corals of American Samoa. It's mostly a simple list of species with some annotation of habitat. But the one on the right is a really robust checklist for the checklist for Hawaiian fishes by Bruce Mundy. And so you can see here, without getting in all the reading, but there's lots of, there's the synonyms of these species, there's geographic information, there's taxonomic information, there's any kind of information you want to put in here can be put on here. And Bruce Mundy's is one of the more robust types of checklists out there. So I kind of aim for developing a checklist along that kind of model. And so how did I develop it? I pulled together every reference I could find, ultimately about 51 different references that I compiled over 46,000 stony coral reports. I recorded all the information that I could associate with each and every report, the type of evidence that supported the observation, whether it was a first order observation or a second order reference, the island it was present at, the depth if it was reported as a depth, or any other kind of information that I could glean from the report. I then used R and WORMS, which is the World Registered Marine Species, to validate taxonomically accepted names and determine which ones were synonyms. And then finally, I took all of that and I produced a final checklist with R and Markdown. This allowed me to essentially write the text in R Markdown without writing 100 and some pages of very dry referencing material and annotation. So with a platform of R and R Markdown, I was actually able to automate the generation of that first draft. And just to jump to the punchline of what this checklist kind of resulted in is there were 745 different names reported within the territory for corals. 541 of those were actually valid species. Of those species, there were 377 actually currently accepted taxonomic names. And of those, I was able to determine 251 is present and 91 is possibly present. 20 is uncertain. 9 as not likely present, and 8 as not present. And so I developed this kind of 5-scaled system based on the levels of evidence of and the certainty in which you could make a determination whether a species is really present or there's some uncertainty associated with that. So I kind of lean on the present, possibly present as the number to work with, and came up with a total of 342 species. So remember, not too far from the 337 that I mentioned just a few moments ago. While there were some species difference, the numbers are quite similar. 90 of which had been where there were depth reports associated with that name. 90 of those were listed below 30 meters. That's about 26% of the species documented. And also I was able to determine 66 of those species were geographic extensions based on Charlie Varon's ecoregions. And of all the work that's been done and so forth, we're able to cross-reference all of those names and all that list with with work that Doug Finner has been doing for many years, and we're actually working with Doug, able to determine 4 of the species that he's found and documented were actually new records for the area. And these are the 4 species that Doug has found within American Samoa, and the checklist was able to confirm that those were the first novel sightings of these species within the territory. But you might remember a few minutes ago, I talked to you about the 6 species that I thought were new records. Once this checklist was fully compiled, I was able to cross-reference those lists and determine that, no, actually 3 of those had been reported previously, but they were not reported in very easily accessible information. For example, a Cropra solitariensis had been collected in American Samoa by Carden Wallace, and there was a specimen in the Queensland Museum, and you would have never known that unless you went to the Queensland Museum data archive and pulled off and did a search within their own database that's available online. So by pulling this information together, we now actually have a fairly robust, dare I say, a complete list of what we know to be in American Samoa. And so just to kind of briefly go through what this checklist is, I'm not asking you to read this slide. I just highlight some of the components of what the checklist is. So if you want to pursue looking at how this is structured, I list here the name, the valid taxonomic name that was accepted. I put a notation here if it was accepted by Varon's Corals of the World or by Carden Wallace's work. I list all the different synonyms that might have been reported that fall under the senior synonym. I highlight what type of synonym it is. And so in some cases, they were just simple misspellings, and I'll get to why that was important in a few minutes. But also I highlighted whether they were heterotypic or homotypic synonyms. And I want to highlight these terms here because these are taxonomic terms that are generally used in the botany world, but I think they apply very well to the zoological community. Zoological community uses slightly different terminologies, but there's been some different, they've been used in different ways, and so interpreting those terms are a little bit hard. The homotypic and heterotypic synonyms are very straightforward. Hetero meaning multiple or mixed, and typic meaning the type. So each species is defined by a type specimen. And so in this case, this junior synonym of a Cropra adani is a heterotypic synonym because this species has its type specimen and a Cropra abertinoides has a type specimen. So we're able to separate out the type of synonym that this is. So we're able to start to pinpoint where the taxonomic issues are or where taxonomies have been collapsed. Homotypic synonyms would just be a species that had basically switched from one genus to the next. So there's still only one type specimen that describes that species, but they're in different genus, different genera. I also make a list of whether they were first order reports, whether they were directly observed, or whether they were referenced. So if a species was referenced to be present by someone else who observed it, I separated those out. I also make a determination, this final determination of whether it was present, possibly present, and so forth. And that's an evidence-based determination. So I recorded the type of evidence. So in some cases, the type specimen of that species might have been collected from American Samoa. So that's the most powerful evidence that a species has been present, even if it's from the synonym. That specimen has actually been well documented. Maybe they're just specimens collected and they were analyzed by an expert. So those were listed. Maybe there was photo documentation that was listed, or it was just an in situ observation that there was no way to validate or verify the observation of that species. So those were all weighted against each other to lead to the determination of whether that species was present or not. I listed the distribution within within the territory. So the island as it's reported, I listed its nearest ecoregion based on Charlie Barron's Corals of the World data. I was able to list its vulnerability status. So in many cases, some of these species are listed by the IUCN. Some cases are listed by the U.S. Endangered Species Act. And then a series of notes. And so these notes can range from anywhere from some sort of ecological observation to an identification issue to taxonomic issues, some sort of confusion that might be associated with that, maybe a recommendation for further study within this group. So anything that was appropriate to tie into this species observation. And then finally, if a species was recorded below 30 meters, it was recorded the depth at which it was recorded and the reference that came from. And so as you can see, it pulls together as much information as we can from all the existing information. And so just a few examples of what kind of things I was able to find here. And so here, the species of Cropra dani also has a synonym called a Cropra acagemensis. And then these two species here, again, it's a heterotypic. So there's two types for these for this species. Cropra acagemensis is a bit of a debated species, and some people believe it probably should not be a synonym. It should be its own valid species. I've looked at skeletons, and I have to agree. I think that there's some validity for that, but the current accepted name is a Cropra dani based on the worm's taxonomy. And so the advantage of using the worm's taxonomy, it's a open source public repository that actually has names and all the synonyms, and it can be a central common point of reference. And so what this does is this points at maybe there needs to be a bit more work. Maybe there needs to be a bit more taxonomic work between these species, maybe even at the locality of something like American Samoa. So maybe the ones in American Samoa are not the same as a Cropra dani that would have been described from somewhere else. So it's used as a way to highlight taxonomic uncertainties. There's also Cropra teres, which is an interesting one in a sense that worms listed this as a taxon inquiritum, which is basically an uncertainty in its name or its status as a species. I decided to elevate this one to as a possibly present within American Samoa based on actual specimens collected both from Hofmeister 1925 as well as Lambert's 1983. Chuck Birkeland found it again in 2007, but it really hasn't been seen elsewhere. And so the thought here is potentially this one's extirpated, maybe not, but we highlight this one to maybe say that maybe we need to be on the outlook for this one. Maybe there's some taxonomic issues, or maybe it's just a super rare coral that deserves a bit more of attention. And then in some cases, there are spelling mistakes. And I'm going to point out some of these, not because I'm trying to point someone makes a mistake, because we certainly all do them. I am no short on that. I make mistakes all the time. But sometimes in taxonomy, simple misspellings can actually have profound consequences. So in this case, there was a Cropra Polyphera reported. A Cropra Polyphera is a ESA listed species in the Atlantic Ocean. It certainly is not in American Samoa, or the Pacific Ocean for that matter. But if you actually dive into it a little bit more, there is a species called a Cropra Polyphera, which is likely what it was meant to be. So it's a simple little typo in 2 letters, simple mistake, but a Cropra Polyphera had been moved to a genus now called Isopora. So it's really, this is probably meant to be Isopora polyphera, but it has no bearing on what the actual names reported. So a slight misspelling can have a big thing. And we're talking about a listed coral that's of important management status. That's a very important mistake that should be highlighted. There's also other confusing aspects like a Cropra caniculata, which is a dubious name. A name does not exist in worms. But there is there's 2 fuzzy matches to this. One is a Cropra paniculata, which is a very close, so it could have been simple misspelling from a C to a P. But there's also a fuzzy match of a Cropra canaliculata. And so Cononliculata is actually a synonym of a Cropra nasuta. I apologize for these names, but I'm trying to tell a story here of how taxonomy and spellings really matter. And so this was probably meant to be a Cropra nasuta. And the advantage here is the National Park Service actually has a photo of this record. And so if you actually go and look at that photo, it certainly looks much more like a Cropra nasuta than it does a Cropra paniculata, which are quite different species. So these spellings, again, can have quite dramatic implications for what the species are that exist. And I'm going to briefly go through another total mess for a coral taxonomy and what the ramifications are of it for biodiversity analysis. And I call it the tale of Samacora. So work by Benzoni et al. Had done a lot of work on morphology and genetics of this group and was able to determine that historical observations of Pavona digitata and Pavona hymiana with an I were actually incorrectly identified. They're actually reversed. So previous to a certain year, 10 years ago or so, I would say, likely these species were probably just simply the wrong name attached to it based on solid taxonomic data and information we have now. There's also a Pavona hymiana with an E, and up to 1976, the Pavona hymiana with an E was likely just a simple misspelling of Pavona hymiana with an I. But corals are never that easy. It's never as simple as a simple misspelling. Turns out, based on the work by Benzoni et al., again, the Pavona hymiana with an E was actually meant to be, it's more taxonomically appropriate to call it Cropra profundicella. All of those species here on the right, hymiana, digitata, and profundicella, are all in American Samoa. There's also another one, Pavona folliculum, which is actually a synonym of Pavona hymiana with an I. And so in reality, all these species are present within American Samoa, but it's quite a complex, messy, it's a messy process to get to that determination. A little bit back to the surveys that were done to pull this back into the deep reef refuge hypothesis, now that we have a good understanding of where we are on all the taxonomy of things. So again, we did a handful of sites. I was able to compare this data with 4 other sets of data. So work done by Maragos in '94 and Coles in 2002, and a whole bunch of sites conducted by Doug Finner. And all 4 of these sites actually are very similar in methodology, survey efforts similar, so they're very comparable in that way. So we're able to take this data and do a species accumulation curve and kind of predict and extrapolate how many species that maybe we haven't found that haven't been observed. And so based on these 4 sets of data set, based on these 4 data sets, able to extrapolate out to estimate there's about 343 to almost 400 species to be found in American Samoa. Now, remember, I just mentioned there was 342 species already documented. So this is actually quite congruent with that. And as we do more work in mesophotic depths and other habitats that we will find more as we resolve cryptic diversity, it certainly will go up a lot more. But you'll see here at the mesophotic depths, since the sample size is quite small, we can't actually predict that far out. But certainly a lot of these new records that may be found will likely come from mesophotic depths. If we were to look at this from all the different diversity metrics things, before I get into this, I'll give a brief overview of what these diversities really are. There's the gamma diversity, alpha diversity, and beta diversity. And essentially what these are is gamma diversity is the number of species within a region. Alpha diversity is the diversity at any particular location, a particular site. And the beta diversity is essentially the difference between those two. There's lots of different ways to define and calculate beta diversity. It's a little bit more complex. But the gamma diversity within these 4 studies, again, shows a vast majority of the species that we know exist to be present within this study. The individual site alpha diversity, the species richness, if you will, at individual sites between the MCEs and the shallow coral reefs, quite similar, except the shallow coral reefs have a much higher variance and distribution of sites. In some cases, upwards of 120 species are found at a single site. That's an extraordinarily high diversity for corals. And keep in mind, Hawaii only has about 60 or so species in an entire archipelago. For the beta diversity, again, there's a number of different ways to calculate that. I use the beta part package within R to use this. And the advantage of this calculation technique is it allows you to actually differentiate with the different components of the beta diversity. And the different components, there can be different patterns that lead to a beta diversity calculation. And so some of that pattern can be the turnover among species. So you have different species that are popping up in some sites and not in other sites. And then you have a nestedness, the species that are found within all sites. And so there's the species identification can lead to these two components that add to the total beta diversity. And the most important part here is that the turnover rate for corals in American Samoa between the mesophotic coral ecosystems and shallow coral reef is quite high. It's majority driven by turnover. Very little nestedness across all these sites. If we were to look at the total community analysis at a multivariate kind of scale, looking at the data from a non-metric multidimensional scaling plot, you can see here there's a lot of all the red triangles are shallow sites, and the blue triangles are the deep sites. You can see there's a lot of variability within these shallow sites. But when you look at the centroids of these sites, it's clearly different, and this shows very statistically different between these communities. But again, there's a lot of variability within this. So that made me ask the questions: what are the types of habitats within shallow coral reefs that actually might have much higher affinity with mesophotic coral ecosystems? That will start to provide more clarity on the deep reef refuge hypothesis. So if we take all the shallow coral reef sites and we split it up by a habitat and island kind of a matrix, we can see here that the upper mesophotic coral ecosystem on the island of Tutila is right about the mid-range of what you would see across all the other different habitats across all the different islands. Some are going to be less, some are going to be more, some have even more variability. But there's not necessarily a single island or a single habitat that ranges just in the total number of species and the total species richness. And if we were to do the same thing with the beta diversity, these four graphs simply show, again, the mesophotic coral ecosystem compared to four different habitat by island sets of sites. And you can see here, this is also driven by the high turnover rate. But maybe slightly interesting here too is that as you move from the reef slope up into the reef flat, so you're moving in quite dramatic habitat differences, you do see even a slight uptick in the amount of turnover rate. So this is something you'd expect. You would expect the reef flat to have a much different community than the reef slope. And if we were to, again, look at this at the community level with looking at all the species across all the sites with the habitat by island kind of approach, you can see here the upper mesophotic coral ecosystem is quite distinct from any other type of kind of habitat by island type of community. You can see here in circled, these are essentially the reef flat, and these are the reef slopes. And then the sites that had combined reef flat and reef slopes are kind of in the middle. So again, these communities are quite distinct. And this is shown, this is statistically significantly different, as well as the dispersion among these sites is significantly different. The dispersion among the sites are not statistically different, which then provides evidence that the community, the centroids of these sites are different. But I will just highlight here that the communities are clearly different, and so there will likely be no replacement community for community A to community B. But there are still a number of species that are overlapping. And so if we really want to dive into the granular questions of a deep reef refuge hypothesis, we really have to start looking at things at a species level. I can show that the communities are different, but what about individual species? And so when we drop out all the unidentified species within the data, we have about 284 total species. 63 of those are shared across shallow and deep, and 209 of those are just in the shallows. And as you can see here, without going through all those names, I just want to highlight here that some species are going to be more common in deeper water and less common in shallow water. Some species are going to have somewhat similar abundance across deep and shallow sites. And then there are some sites or some species that are found in deep water, but they just simply have not been found, or in maybe in other studies, extraordinarily rare in shallow water. So taking the concept of abundance in mind or commonness in mind, I started to ask the question is, are we really looking at the deep reef refuge hypothesis really in the right way? And so previously, studies have talked about generalist species and specialist species. And so generalist species are species that are found between both depths, and specialist meaning only found in one or the other. But that's a bit kind of broad to really start to look at these questions. So I decided to propose to kind of redefine what we mean by specialist and generalist. And so to do that, I broke up the data set into what I would call common species, occasional species, and rare species. So common species are, in this case, species that are found about 50% or more of the site. So it has a 50% or greater site occupancy. And the rare species would be 25% or less for site occupancy. And then the ones in kind of in the middle are more occasional. So they could kind of go either way a little bit. Now, this could actually be applied to data that actually has density or coral cover or other types of density metrics. I had presence absence data across sites, so I was able to use a site occupancy kind of approach. And so if we were to do that relative comparison, so if they're found common in one depth and less common in the other, we're able to kind of categorize the species. And so if you kind of look at it this way, we can come up with what are common deep specialists or shallow specialists. We can then go a little bit more granular detail if there may be occasionally specialist. But maybe of most importance here would be to look at the species that are common or at least occasional across both depth ranges. And so in this case, there are only 6 species of all of these that are actually common in both mesophotic depths as well as shallow coral reef depths. I've also done this analysis across all the other 14 habitat by island groups. And so if we were to do that same kind of analysis, there's actually about a total of 19 species that would fall into that relative comparison. And so I look at potentially about 19 species that might be of more interest for a deep reef refuge hypothesis. Of course, we always have a whole bunch of species that are rare in communities. And so in these cases, both these species are rare. These species are rare in both deep and shallow. So I call these rare generalists. And of course, we still have to highlight the species that are not found in both deep and shallow. And so in this case, this is showing the deep exclusive species. There would be a whole bunch of shallow exclusive species that I'm not showing here. And to kind of put this in the context of what it actually might mean for the deep reef refuge hypothesis, we're going to have some species that are shallow exclusive species and deep exclusive species. And so clearly those, we can throw those away. Those wouldn't serve much of a refuge. They're only found in one depth, so a disturbance event that would occur across all these habitats would simply not work. But if we had common or occasional specialists, they're more common in deeper water, less common in shallow water. Here I propose the concept of looking at both rarity and specialty as an issue to consider within the deep reef refuge hypothesis. So if a species is a specialist or is rare, it's likely to serve a far less likelihood to be a potential reseeding species. Remember, I talked about reseeding species, the possibility of the propagules from deep to shallow. And so if you're a specialist in deep water, you may not do very well in shallow water. So you're not necessarily prone to be a good recovery potential for shallow water impacts. And the same could apply for the shallow specialist. You're very rare in deep water. You're gonna have a harder time being to repopulate or to supplement an impact to a shallow coral reef. And of course, there's a common and shallow occasional generalist. So if you're found commonly across all depths, you're gonna have a little bit more robustness, maybe a bit more resilience in order to provide as a reseeding ability to the shallow coral reef. So if you have a certain habitat that's impacted more than others, you have certain depths that are impacted more than others, you're going to have a potential higher likelihood of being a receding species. And then the rare generalists, again, if you're just rare, if you're a rare within a habitat, you might be more of a persistence role. So the species may have a less chance of going extinct or extirpated, but it's going to have a lower possibility to develop to be a reseeding or a refuge type of species. And so again, 19 of those species I determined to be good potentials for reseeding species to serve as a refuge. That's about 25% of the MCE species, but only about 7% of the entire coral reef, shallow coral reef community. Now, if you want to push back on me and say, well, you know, if you're found in both habitats, you can still serve some sort of potential for reseeding refuge. Again, remember, there are only about anywhere from 63 species, or if you want to look at the checklist data, 90 species, you're only looking at about 23 to 26% of all shallow coral reef species that have ever been found in the mesophotic depths. Now, I will highlight also that this is from the upper mesophotic zone. Once we actually go deeper, there's a much higher likelihood of having even further unique communities, further distinct communities. And so the mesophotic zone likely will have as many or more subhabitats as the shallow coral reef community would have as well. So just by simply looking at the species overlap and the amount of that, there seems to be a very small chance for the deep reef refuge hypothesis to be any kind of meaningful ability to recover a shallow coral reef, even if it does pick out, even if it does work for maybe a small handful of species. And just to bring in a little bit some of the take-home messages today, MCEs account for about 79% of all coral reef area between 0 and 150 meters. And that's 86% on just the island of Tutuila. The MPAs protect about 12.5% of the coral reef depths, and some of those in quite high percentages protect MCE depths. There are about 342 coral species that are present or possibly present in American Samoa, but they're likely upwards of 50 or more species yet to be found, based on if we had an infinite number or hundreds more of survey samples. And of all that being found, there were 3 species found that in mesophotic depths that are actually listed under the Endangered Species Act. Cropra speciosa being an occasional deep specialist, Fimbrophilia paradivisa being a deep exclusive specialist, although it had been found in other surveys in shallow water, so maybe it's a rare generalist. Or Pavona diffluens is a rare generalist. The coral communities across different reef habitats and islands are distinct. There is no simple replacement, even at a smaller habitat level. But despite the species overlap, there are likely only about 19 or so species that could have a good potential for reseeding species, if we take into consideration the concepts of rarity and specialty among habitats. Even fewer of those species are likely to actually serve as a reseeding species once we actually go through the complicated process of documenting their ability to have reproductive capacity at depth, their ability to recruit across depths, their genetic connectivity, clarifying any kind of cryptic diversity, which seems to be more and more common along these communities, and the fact that are these, even if they were able to reseed a shallow reef, do they have the genetic resilience or the ability to withstand the same level of shallow coral reef disturbance events? So the MC communities likely do not provide any substantial reseeding ability, and the MC communities are really should be considered unique, and they're really a broader continuum of the coral reef ecosystem. So we really should be talking about coral reefs between 0 and 150 meters, not in the upper 30 meters that we're more familiar with. So I would say that the MCEs preserve their own protection and consideration for conservation planning. Let's not look at these as a fail-safe or as something that's going to help our shallow coral reefs. Let's treat them independent. Let's treat them and protect them for their own biodiversity and uniqueness and ecological needs. The deep reef refuge hypothesis may also work in reverse. So remember, we're talking about from deep to shallow, but often the conversation between shallow and deep gets forgotten. And so there very well may be cases where shallow coral reefs are actually supporting mesophotic coral reefs. And so this should not be forgotten in the broader context of looking at coral recovery and impacts. And with that, there should be a broader framework for considering the recovery. So instead of asking developing the hypothesis of this broad do deep reefs support shallow coral reefs? What habitats are better connected with others, and what kind of recovery potential are we having at a finer scale among these communities? And for the most part, deep coral reefs just do not appear to work for most coral species. So just based on numbers, there are far more species in shallow coral reefs than deep coral reefs. And so even if they did work 100% of the time, that's only going to be a small fraction of the total diversity. And just to make a couple points on conservation planning, while American Samoa has a pretty good system of marine protected areas compared to many other areas, I would argue that areas as they get developed or refined should include more MCE explicitly, much like the National Marine Sanctuary of American Samoa has done. And so looking at the habitats in the habitat diversity in developing these areas should be a consideration. And also will, as a final note, National Marine Fisheries Service is now currently proposing critical habitat for these listed species. And in that proposal, they're limiting the depths to 40 meters. But as I've shown here, 3 of these species are actually found below 40 meters. And so that might be something worth considering, is should critical habitat only be limited to 40 meters? As offshore projects may get developed with renewable energy or cable alignments that cross these habitats, maybe critical habitat considerations should be included below 40 meters. And with that, I just want to take a couple minutes before we take some questions and just give some people, I can't thank everyone, but there are certain people I owe a huge debt of gratitude. So a person in my status, I've been in the field for a few years. Everyone I've worked with has a role that I can thank. I'm not able to do that here today, but I really have to start with my committee, all 5 members of my committee, both Rob, Megan, Randy, Celia, and John. Nothing but supportive from day one. And I truly, truly appreciate all the advice, guidance, and support you've given me over the last, excuse me, 7 years. Hard to believe it's been 7 years. I have to thank excuse me, I have water down the wrong way. I have to thank my funding agencies, both the Coral Reef Conservation Program, as well as the Competitive Research Program at NCOS, for the work that they funded. Certainly I wouldn't be able to do a lot of what I've done without that support. Support the Lord Scholarship to HIMB for students. That was a huge help. And while my office of the Pacific Islands Fish and Wildlife Office hasn't actually supported funding, they have been hugely supportive of my work. And so I have to thank my supervisor, Dan, my other supervisor, Greg, for the support they've shown me over the last several years. Working full time and doing this on the side has not been easy. And so without their support, it wouldn't have been possible. I also want to note within the Coral Reef Competitive Research Program, I have to give a huge thanks to Kimberly Pugliese. She's been a huge supporter of MCE research over the last well over 10 years now. A huge supporter of work in American Samoa. So I thank her for the support she's provided to not just the coral reefs, but to me personally as well. I can't thank Doug Finner enough. He's been a huge resource for me. Both in coral taxonomy, a sounding board for all things coral reef. Without his participation and support, no way I would have been able to do what I've been able to do. So Doug, I hope you're here. I thank you very much. I appreciate everything you've done. I can't help but to thank Dave Pence. Dave, you've been great. I really, really appreciate all the support you've given me, but maybe more importantly, everything you do. The program you support at the university allows this type of research to go forward. So I thank you for not just the help you provided me over, I think it's been 20 years or more now, Dave. I appreciate that. I thank you. I wouldn't be here again without your help. I thank you immensely. My thanks can't be enough. I have to thank Richard Pyle. I've known Rich for more years than I'd like to say. Rich has been a friend, a mentor. He's guided me in a lot of ways. He's helped me in a lot of ways through this dissertation, through a lot of advice. He's always been available for bouncing ideas off. I'd just like to say I wanted to share this photo of everyone because it's proof that Rich does more than just fish. Here he's collecting some corals. So, hey, Rich, there's always a place in the coral community for your help. So please come join us. And I have to thank these two gentlemen too, both John Rooney, as well as Josh Kopis. Unfortunately, they're not with us today, but they both played a huge impact on me. And I thank you for that. And finally, sorry, I'm a little emotional here with the family, but both my wife and my two daughters, thank you very much for everything you've done. I can't really adequately explain the thanks, but you deserve this as much as I do, so thank you. And with that, I'll take some questions. Thank you.
Let's go ahead and unmute and collab. Yeah.
Well done, Tony.
Well
Round of
done.
applause.
Well done. Way to go. Well deserved, man.
Way to go.
Yay, Tony.
Way to go.
Beautiful work.
Great job, son.
Congratulations, Tony.
Thank you. Thank you, everyone.
Great job.
Tony.
All right, and with that, if you would like to ask questions, please go ahead and unmute, or if you want to leave them in the chat window, that's fine too. I'll try and pass those along to Tony.
Can I ask a question?
You may.
Thanks. Hey, Tony, it's Matt. Fantastic amount of work and a massive amount of effort involved. How do you see this kind of melding with management styles? You know, could we use, I mean, that's sort of what everybody's wrestling with is a deep refugia viable as sort of a management alternative? And obviously you have a better understanding of it than I do. So I'm just curious, is that something that we can count on, or should we really be putting our eggs in another basket?
Yeah, well, thanks for that question, Matt. I think your question really gets at the heart of it. I don't think it is something we should be counting on, even if it was to work in even more cases than what it probably does. You know, we really need to treat these systems are a continuum of the broader coral reef ecosystem. But just like between reef flats and reef slopes, or atoll reefs and high island reefs, or even reefs that are spatially separated apart, we have to maintain the diversity within our management regime. And so we don't want to rely, we wouldn't want to rely on the reef slope to protect the reef flat, right? So we wouldn't want to do the same thing for deep reefs as well. So we should manage them separately. We shouldn't rely on them. And to how do we apply that in the reef in the management community? That's an extraordinarily difficult thing to actually achieve. But I think whenever there's a management action that needs to be taken, whether that's developing an MPA, whether that's something as narrow as Endangered Species Act action that has to be taken, whether that's even a fishing regulation or any other type of management action needs to be considered, we should think about the mesophotic habitat to its own right and to its own resilience within itself, and not rely on some sort of associated or other community that could support it further.
Yeah, that's a good answer. I appreciate it.
I have a quick question. Thank you so much for that talk. I was curious, in your opinion, so the kind of 6 or so species that you found that were common generalists, do you feel like there's any uniting characters or something about those species in particular? They're obviously very phylogenetically diverse, taxonomically diverse, but do you feel like there's anything that unites those species in your opinion?
Yeah, that's a really good question. To be honest, I hadn't thought too much about that aspect, but the thing that has stood out to me and stands out to me now is that they are really just common species. I think one of them, if I remember correctly, was Leptastria purpurea. I mean, the species is found everywhere, right? And so it could just be the species that are much more resilient. They're much more genetically diverse to be able to adapt to varying light levels, temperature regimes, a wide diversity of environmental variables. And so that would be the first place I would want to look exploring that question, but I hadn't really explored it at that level. So that would be my best estimation.
Great, thank you.
This isn't a question that relates exactly to what you were studying, but I'd be curious, corals can live a lot deeper than they can make reefs. Did you, in all your experience, see where the Darwin point, where the corals are actually accreting the structure and where they're just living on other rocks and things that are down there? Or did you not? It's not a fair question in that wasn't what you're after, but I'd like to know your perception since you had the experience.
I think it's a very fair question. And the answer is quite simple, is that the work that I've done is in American Samoa has only been 30 to 70 meters. So you wouldn't see that in the depth zones, in the survey data that I have. But I really, really appreciate your comment and your question by pointing out the Darwin point, because that's something that has not been brought up in the vertical distribution of coral reefs as much as it should. And so I don't think there's an easy answer, whether you're in American Samoa or Hawaii or anywhere else. And there likely is a point. Whether these reefs actually even are accreting at these depths at all is a question, let alone is there a break point at which that changes or not. I think that's a much bigger question. And so easy answer is I didn't really observe it because I didn't look for it. I wouldn't know it even if I probably saw it, but it's an extraordinarily important question and consideration that we need to think of.
Tony, there's a question in the chat asking if you think threats are manageable by MPA or are they mostly global change issues?
Well, that's a really good question. I think there's going to be a combination of both. So I'll handle the climate change aspect first. I don't think we truly understand what the impacts of climate change is going to be on mesophotic coral reefs. There's certainly some evidence that they're going to be a little bit more insulated from warm water bleaching events than shallow water. Based on some of the work, but that doesn't mean they're fully protected against that for a number of reasons. So even if bleaching events would extend down deeper, the species might even be less resilient than shallow coral reefs, right? So they could be actually more vulnerable than less vulnerable. And then there's aspects of storm pattern changes and so forth. There have been documented evidence of storm damage deeper water, which is not something that maybe had historically been thought of to truly exist. On steeper slopes, there can be landslides that come down from storm events. There can be all kinds of other storm related aspects. And so what the impacts of a climate change under different climate change scenarios could be is hard to predict. But short of managing carbon sequestration and discharge, all we can do is at the local level. So whether that's maintaining our resilience among all our coral reefs, including mesophotic coral ecosystems, through marine protected areas, or in some cases, if there are fishing pressures that are causing impacts, or there are other types of things that we consider. I think we have to start at the local level for our management. And then let's hope that the more broad scale regional or global management can catch up in time. I'm hoping I answered that question thoroughly enough.
Are there other questions? There's a follow-up. What about bottom fish anchor damage?
Yeah, I don't, I've never, the limited work I've done, I've not seen that, but certainly if bottom fishermen are anchoring, you don't know what you're anchoring onto. And if you're looking for bottom fish, you're going to be looking for those kind of habitats that have some rugosity, some hard bottom, somewhat in the vicinity. So there could be some attraction to that. And so if you're anchoring, certainly that would be a small scale impact that should be looked at.
All right, any other questions?
Hi, Tony. Kim Fleur here. Great presentation. I was wondering if you saw any aquatic invasive species at this depths, or if that could also serve as a refugia from aquatic invasive species.
Well, the simple answer is no, I did not, but I wasn't looking for them. So my focus was on corals, and if I saw a fish, I'd be surprised. So I didn't see anything. Given that my identification skills in American Samoa was quite limited, I don't know if I would recognize it if I saw it anyway. Keep in mind the diversity in American Samoa compared to Hawaii is nearly an order of magnitude more. So that's a challenging aspect that I wouldn't be able to do.
There's another question in the chat asking if you have any thoughts on the previous mortality that you observed.
Yeah, well, good. I was wondering if someone's going to ask me that question. I didn't want to spend too much time talking about it, but I was on a series of sites that were very similar in nature. So they're offshore patch reefs. They were somewhat on different sides of the island. Many of them were on the north side. And there was a lot of coral structure there, meaning that you could see the plate corals or cropras that were there. The structure was still existent, so it hadn't, but they were dead. And the fact that that structure still existed was an indicator that mortality had not occurred from years and years prior. It had probably occurred whether it was not freshly dead. So there was some coralline algae growth. So it was probably within the last few years at the most. But the coral structure was still there. In addition, we were also observing some Cropra recruits. So there was some recruitment coming into these areas. So we'd find lots of little baby Cropras recruiting to these areas. And so the only two kind of hypotheses I would kind of accurately, or I should say, I feel like I could defend was whether there was a disease outbreak that had quite a mass kind of, I should say three. There could be a potential disease outbreak that had mass mortality. There could have been bleaching events at these depths. Or American Samoa, particularly the years prior to this, this was 2016, actually had quite a bit of crown of thorns outbreaks. And so based on some discussion with other people there, a lot more local knowledge, some of these northern areas were hit pretty hard. And there is a hypothesis that some of these crown of thorns were coming from deeper water. So it's possible maybe they were recruiting onto these little patch reefs. There's not a high coral density there to begin with. So they gobble that coral up and eat it, and then they move on and migrate elsewhere. So the crown of thorns is I would say maybe the leading hypothesis if I had to guess, but we couldn't rule out a disease or a bleaching type of impact.
Right, I think we have time for about one more question before I'm going to ask everyone to exit and let the committee have their chance to talk to Tony.
Yeah, I'd like to ask Tony, first I want to say what a nice job you did. This really great, and I certainly appreciated the time that you spent dealing with the taxonomy, which I think is a very important thing to do. But I also want to ask whether the widespread species that you had, I think there were 19 species or something like that. Do you have any sense to what extent those might be pseudo populations? In other words, organisms can settle and maybe even grow in some places, but the conditions are not favorable enough for reproduction. So if they can't reproduce, then they're not going to do any reseeding.
Yeah, I think it's a great question. It really hits at the heart of what I'm kind of trying to talk about here, is that if you're a rare species, there might be more likelihood that kind of model fits. Maybe it's the peripheral of your ideal habitat or environmental parameters. There likely could be very real issues with cryptic diversity here. Something like some of the leptastrias, things that are, or even some of the others that are found in such extreme habitats. It's not impossible. They're actually just different species. It's just, we can't visually simply identify it. Certainly cryptic diversity is popping out in a lot of scleractinians right now. That could diminish those 19 species even further. If you're a rare specialty kind of species, you're going to have less likelihood to be able to maintain that population. And so they very well aren't connected. And so again, that wasn't part of my research to start to look at this reproductive output and this genetic connectivity and all the other aspects, but certainly those 19 species could be the ones that you would target to then get at those questions you're asking about. If we take a step back and say, okay, 19 species out of 300, well, that's not much of a refuge from a community level. Maybe we hold on to a few species, but in order to hold on to a few, there's a long list of things that species has to be able to do to actually serve as a refuge. And so the 19 that I'm proposing as a possibility likely will go down quite a bit. And the work that's been done on the deep reef refuge hypothesis at the species level, some have shown support for it because there's reproductive output across habitats, and others have shown they're not. Maybe the spawning and the reproduction is asynchronous. Maybe there's not genetic connectivity. The genetic structure is just different. In some cases of Ceratophora, there were clearly cryptic species found within these communities. So I think it's an uphill battle, even for the species that may have a possibility.
Yeah, great, thanks.
All right. Well, please join me once again in a round of applause, and I'll ask the committee to stay afterwards.
Yeah, no.
Congrats, Tony.
Done, Tony.